In Chapter 3, we're going to try to understand how microscopes work, the tools, the primary tool that microbiologists have to work with, and the tools that we'll be using, especially light microscopes, and then look at staining and how we can use staining to make it easier to visualize the things that we want to see in the microbes we're looking at. So, let's get started on. As we learned in last chapter, it was Anton van Leeuwenhoek who built the first quality microscopes and was able to magnify things visually by about 300 times and was able to look at a lot of stuff this way. So he produced very high quality lenses, mounted them here. Dadgummit, I can't. Let's try my arrow again. Okay, so here we go. There. Mounted right there in that little spot, the lenses that he made. And then he could use the positioning and focusing screws to get the subject of what he was looking at into position and then move it back and forth closer to or further away from the lens in order to be able to get it focused and look at it well. And he produced a lot of really amazing drawings from the work that he did. So when we think about the sizes of the organisms that we'll be looking at in microbiology, we measure them in millionths and billionths of meters. So a meter is approximately, you can think of it as a little bit more than 3 feet. So like 3 1/3, give or take, feet. And when we talk about microorganisms, we usually use micrometers or microns and nanometers, sometimes called millimicrons, but we'll use mostly the terms nanometer and micrometer. A micrometer is 1 millionth of a meter, so a millionth with an M of a meter, while a nanometer is a billionth of a meter. And so if you hear people talk about nanotech, they're talking about things that are the size of 10 to the minus 9th meters, or 1 billionth of a meter. And then last of all, the angstrom is used primarily when talking about atomic sizes, so atoms and so on. And that's 10 to the minus 10th meters, or 0.00000000001 of a meter. So, there you've got it. Those are the units that we'll be talking about. So clearly the stuff is super, super small. We also need to have some introductory information about light and a little bit about how vision works. So we see objects because light energy reflects from these objects. And it depends on the wavelength of the light energy to determine what the color is of the object that we're seeing. And so let's say you're looking at a blue object and you're looking at it under sunlight, then you're getting all of the wavelengths of light coming, being beamed at whatever that object is. But as the red, the orange, the yellow, the green, the violet wavelengths meet the object, their energy is absorbed. And the only energy that's being bounced back or reflected from the object then is the light energy that's in the blue wavelength. And so it appears blue. And in the same way, if it's a red object, the pigments in it absorb all the other wavelengths except for red, and it reflects red back to our eyes. And our eyes bring in this information, so here comes the red wavelengths, and when they meet the retina at the back of the eye, the cones of the eye, remember that from a previous course hopefully, there are cones and rods. Rods see the difference between light, the amount of light, so the rods are noticing whether there's lots of light or very little light. We think of that as kind of black and white vision, while the cones are noticing what the wavelength of the light is that's coming at them. And so if it's red, then the red cones are going to be activated and we see a red image. It's the wavelength of light, and that is the Greek symbol for wavelength. It's lambda. Lambda is the wavelength. Now there's some interesting things about the way that light works though, and this image is missing some important stuff, unfortunately. So if light is hitting a white object, what is happening is that what's reflected back are all of the wavelengths of light. And so as all those wavelengths are reflected back toward the eye, what we perceive as the three different types of cones are activated. So we have red, green, and blue cones. They're sensitive to those wavelengths primarily. But as those different cones are activated, what we wind up seeing is white light. We perceive white as the color that's being of the light that's coming back at us when we look at white things. On the other hand, if all those wavelengths are absorbed and there is no light being reflected back to the eye, then that is perceived as black. So if the red, the green, and the blue cones, none of them are being activated as we look at stuff, then we see that as black. So if all three of them are activated, then we see white. If none of them are activated, we see black. Okay, so what is this light radiation that we're talking about? Well, it's just one small segment of the electromagnetic radiation that is produced, the energy radiation that's produced in the universe. So energy travels as waves, like this. So a packet of energy moves as a wave. And what we talk about as the wavelength is the distance from one peak to one peak. So there's the wavelength. And it's also the same as saying one valley to one valley. Those are the same distances. So that is the wavelength of light, that distance from peak to peak or valley to valley. If it's really short, so less than 1/100th of a nanometer, okay, so super short, then though that energy is described as gamma radiation. So gamma radiation is radioactivity. So are X-rays. So X-rays are produced as radioactive materials break apart. And so gamma rays, X-rays, they have super, super short wavelengths. The next longest set of wavelengths are what we think of as ultraviolet light. UV light is, it's not visible to humans. There are some animals that it's visible to. So insects have reasonably good vision in the ultraviolet spectra. But for humans, it's invisible. But short wavelength relative to visible light. It's shorter than what we can see. And then we get to the range of what we think of as visible light, the range of electromagnetic energy that will stimulate the cones and the rods of the eye. As the wavelengths get longer, then we get to the infrared rays. So if you're at McDonald's or whatever fast food place you like, and you're seeing the fries being kept warm under those kind of orange-colored lights, that is infrared light that they're emitting. So you're seeing some of this near infrared radiation that's visible to you, but there's also a whole lot of radiation that's coming in the form of heat or infrared radiation. And then we get to microwaves. So those are wavelengths that are capable of interacting with water and other molecules and causing them to vibrate, producing heat. So microwaves are longer than the infrared waves and longer than visible light. And then finally out at this end, we get to the radio waves, the longest of the wavelengths. These are the wavelengths that can travel far, far distances. So we send them out in space in order to, or we receive them from space in order to see what's way out there. So there's a huge range of energy and electromagnetic radiation. There's a huge range in the wavelengths. But the stuff that's right here in this range is that's the energy that's visible to humans. And if we look at the different wavelengths along the way, the very longest wavelengths are in the violet range, what we perceive as violet. Then indigo is a little bit shorter. I'm sorry, I got that wrong. These are the shortest wavelengths. There we are. Sorry about that. The very shortest wavelengths are in the violet range. And then we get to indigo, which is a little longer, and blue, a little longer yet, and then green, and then yellow, and then orange, and then the longest are the red wavelengths out here. And now, hopefully it makes a little sense why we call this non-visible but near to the violet wavelengths ultraviolet, because they are shorter wavelengths than in the visible violet range. Where infrared, that is longer wavelengths than we see in the visible red wavelengths. So, gamma rays and that end, super short wavelengths. Radio waves, super long wavelengths, sometimes longer than a mile. Those short wavelength energies, they don't travel very far, but they carry lots of energy with them. The long wavelengths, they are much longer, can travel much further distances, even though there's less energy associated with them. Okay, so that is a little bit about electromagnetic radiation, and so some about how we perceive light and make sense of it. So those visible wavelengths, the Roy G. Biv, so violet, blue, indigo, blue, green, Roy, yellow, orange, red. There we are. Those wavelengths are between 400 and 700 nanometers long, or 0.4 to 0.7 micrometers long. Same difference. As we said, the shortest wavelengths are violet, while the longest wavelengths are red, with green and yellow somewhere as intermediates. And when we perceive something as being white light, we perceive it as white because all of our cones are being activated. The red, the green, and the blue cones all are being activated. And when they're all activated, then what we perceive is white. When just red and green are activated, then we see yellow. If red and blue are activated, we see purple. And if green and blue are activated, we see this light greenish blue sorts of colors. Okay, so that's how we perceive color. If there is no light coming to the cones, then we perceive black. And so if we're thinking about looking at something black, again, no light is being reflected from, no colored light is being reflected from that region of this image that you're looking at on your computer. Okay, so the last concept then to get to is around the principles of microscopy, and we will approach the first of these principles, and that's the idea of resolution. So one of the issues with van Leeuwenhoek's microscopes and why he couldn't get beyond just like 300x magnification is issues of resolution. So resolution is the ability to perceive two objects that are near each other as being separate. So for instance, those two dots, you hopefully can see them as separate. If you were further away, perhaps they would blend into one, whereas I'm going to make a dot and then put one right next to it. Oops, and I can't get, let's see if my little thing you can get there. Perhaps you can perceive those two as being separate if your vision is really good, but step back a little bit from the screen and it doesn't take much distance away to begin seeing just one dot rather than two dots. So images that have poor resolution then have lots of objects that could be seen as separate, but they appear in the image as one. And so if you look at these two images of pollen grains, in the bottom a poor resolution image, kind of a blurry looking image, versus a high resolution image. So in order to increase resolution, what you have to do is send shorter wavelength light. So you have to send, or I should say not light, but energy. So the shorter the wavelength, the easier it is for that light to get in between the objects that you're looking at. And so if you want to resolve something where the objects that you're trying to resolve are very close to each other, so if you want to look at really tiny things, essentially, you have to use the smallest, shortest wavelength energy possible. And so that's why first off, that's why light microscopes have a limit to how much you can increase the size or, increase magnification. About 1000x is about the best that a light microscope can do. Electron microscopes, because they're using a beam of electrons that have very, very, very short wavelengths, electron microscopes have the greatest resolving power currently. And so we can resolve things that are as close to each other as 0.2 nanometers. That would be 2 angstroms apart, if you think about the difference between a nanometer and an angstrom. So we are capable now to see with some of these electron microscopes, we're capable of seeing very, very tiny things, of actually being able to visualize atoms and molecules with them because of their ability to resolve. But the best we can do with a light microscope is about 1000x. And that's what we'll be doing. Most of our microscopy work will be at 1000x with these dudes. Okay, so that's resolution. So just to review, we've talked a little bit about light and how light energy is just one small segment of the entire range of electromagnetic energy. And so we use light with light microscopes, but we can use much shorter wavelength electromagnetic energy when we employ tools like electron microscopes in order to resolve very, very, very small structures. Okay, you know how to reach me. If you have any questions, come see me during office hours or send me an email. Talk to you soon.